Chapter 23. Rebuilding Sequence. Rebuilding the digestive sequence requires restoring the order, timing, and dependency relationships that define how digestion operates as a system. Each phase prepares the next. When that order is disrupted, downstream processes are forced to operate on substrates that do not match their requirements, and compensation begins to replace coordination. The objective is not to correct isolated failures, but to re-establish the chain of preparation so that each step receives the correct input at the correct time. Sequence is the mechanism that converts intake into usable output. Without it, digestion becomes fragmented and inefficient. The starting point is the re-establishment of early phase signaling. Sensory input initiates the cascade that drives gastric and pancreatic output, and this input must be present and sustained long enough to generate a full response. When this phase is restored, the stomach enters a state where acid production rises in proportion to the incoming load, creating the conditions required for structural modification of proteins. This step determines whether downstream enzymes will be able to act efficiently. If proteins remain folded and resistant, the rest of a sequence inherits that limitation. Gastric processing defines the structural state of all incoming material. Acid exposure unfolds proteins, alters physical properties of the food matrix, and sets the rate at which contents are released into the small intestine. Rebuilding sequence at this level means allowing gastric conditions to develop fully before progression occurs. When this is achieved, the material entering the intestine is already partially processed and aligned with the requirements of enzymatic breakdown. When it is not, the intestine receives material that is structurally incomplete and processing capacity is exceeded. The transition between the stomach and small intestine must be resynchronized so that delivery matches processing capacity. Gastric emptying is not a passive event. It is regulated based on the condition of the contents and the readiness of the intestine to receive them. Rebuilding this transition means restoring the feedback signals that control release. When these signals are functioning, substrates enter the intestine in controlled amounts that can be matched by enzyme secretion and bile flow. This prevents overload and maintains alignment between delivery and processing. Pancreatic enzyme release and bile flow operate as a coupled system within the intestine. Enzymes reduce macromolecules into smaller components, while bile organizes lipids into structures that can be accessed and absorbed. Rebuilding sequence here requires that both systems activate in proportion to the incoming substrate load. When they are synchronized, breakdown proceeds efficiently and absorption can occur without accumulation of partially processed material. When they are not, incomplete substrates move forward and downstream regions compensate through microbial metabolism. Absorption and transport complete the sequence by moving processed substrates into circulation and distributing them to tissues. This step depends on the prior phases having produced the correct molecular forms. Rebuilding sequence ensures that transport systems receive substrates they can handle, allowing for efficient uptake and delivery. Lipid transport, in particular, provides a sustained flow of substrates that stabilizes energy availability and supports consistent signaling across systems. The rebuilding process also requires removing patterns that disrupt sequence. Rapid intake, inconsistent pacing, and irregular timing compress phases that should remain distinct. This compression forces multiple steps to occur simultaneously without proper preparation, weakening the entire chain. Restoring sequence separates these phases again, allowing each to complete its function before the next begins. Rebuilding sequence therefore reestablishes digestion as an ordered system where structure leads to function and function leads to output. Early signaling prepares the system. Gastric processing modifies substrate structure. Controlled release aligns with intestinal capacity, enzymatic and biliary activity complete breakdown, and transport delivers nutrients in a stable pattern. When these steps are restored in order, digestion returns to a state where each phase supports the next, and the system operates as a coordinated whole rather than a series of compensations. Acid bile enzymes motility. This sequence defines the operational order of digestion, where each phase sets the conditions required for the next to function correctly. It is not a list of independent components. It is a dependency chain. When the order is preserved, digestion proceeds with coordination and efficiency. When it is disrupted, downstream processes receive substrates that do not match their requirements, and the system shifts into compensation rather than alignment. Gastric acid establishes the structural state of incoming food. Proteins are unfolded, connective structures are loosened, and the physical matrix of the meal is altered in a way that increases access for downstream enzymes. This step determines whether enzymatic cleavage can occur efficiently. When acid exposure is sufficient, substrates are reduced into forms that can be processed further. When it is not, larger and more resistant structures persist, limiting the ability of enzymes to act and setting a constraint that carries forward through the entire sequence. Bile follows as the structural organizer of lipids. Once gastric processing has reduced and dispersed the meal, bile interacts with lipid components to break them into smaller, uniformly distributed structures. This increases surface area and allows enzymes to access and act on fats effectively. Without this step, lipids remain in larger aggregates that resist enzymatic action. The presence and timing of bile therefore determine whether fat processing proceeds efficiently or becomes a limiting factor in the sequence. Enzymes act on substrates that have already been structurally prepared by acid and bile. Proteases, lipases, and other enzymes require access to specific molecular configurations. When upstream processing has occurred, enzymes can rapidly reduce macromolecules into absorbable units. When has not, their activity is restricted and breakdown remains incomplete. Enzymes do not compensate for poor preparation. They operate within the constraints defined by earlier phases. Motility governs how substrates move through the sequence and whether each phase has sufficient time to act. Coordinated motility patterns mix contents, distribute enzymes and bile, and regulate the rate at which material progresses through the system. When motility is aligned, substrates remain in contact with the components required for processing, and each phase completes its function before progression occurs. When motility is disrupted, material may move too quickly, reducing contact time, or too slowly, increasing exposure to partially processed substrates and altering the internal environment. The sequence functions as a continuous flow where structure determines access, access determines breakdown, and breakdown determines absorption. Acid prepares, bile organizes, enzymes cleave, and motility coordinates. Each step depends on the one before it, and failure at any point alters the conditions for everything that follows. Rebuilding this sequence restores digestion as an ordered process rather than a fragmented one. When acid production is sufficient, bile flow is coordinated, enzymatic activity matches substrate load, and motility maintains proper timing, the system converts intake into usable output with consistency. When any part of this chain is weakened, the effects propagate forward, reducing efficiency and increasing variability in downstream outcomes. Why order matters more than inputs? Order determines whether inputs can be used at all. The digestive system is not designed to handle raw inputs in any form at any time. It is designed to transform them through a sequence where each step changes the structure of the material and prepares it for the next operation. When that order is preserved, even variable inputs can be processed effectively because each phase receives substrates that match its requirements. When the order is disrupted, even high-quality inputs fail to be utilized because they arrive in forms that the system cannot handle. The constraint is structural. Gastric acid must act before enzymes can access proteins, bile must organize lipids before they can be broken down and absorbed, and transport systems can only move molecules that fall within specific size and polarity ranges. These are not preferences. They are physical requirements. If a step is skipped or weakened, the substrate remains outside the range that downstream processes can act on. No increase in input quality changes this. The system cannot bypass structural preparation. Timing reinforces this dependency. Each phase requires a specific window to act on incoming material. When intake is rushed or when transitions between phases are compressed, multiple processes overlap without completion. The stomach releases material that has not been fully processed, the intestine receives more than it can handle at once, and enzymes operate on substrates that are not ready. This creates a condition where inputs are present but not usable, and a portion of those inputs moves through the system without being captured. Transport systems further enforce the importance of order. Absorption is selective. Molecules must be reduced to defined forms before they can cross the intestinal membrane. Larger fragments, poorly organized lipids, and partially processed carbohydrates remain in the lumen regardless of their origin or quality. The presence of these materials does not contribute to the internal substrate pool. They are effectively excluded from the system. When order is preserved, the same inputs produce a different outcome because they are transformed in sequence. Proteins are unfolded and cleaved into amino acids, lipids are emulsified and reduced into absorbable units, and carbohydrates are processed into simple sugars. These forms can be transported, distributed, and used by cells. The system extracts value because each phase prepares the next, and the entire chain operates without interruption. When order is disrupted, the system shifts into compensation. Downstream regions attempt to process material that should have been handled earlier. Microbial activity increases to metabolize what remains, and immune signaling responds to the presence of incomplete substrates. These responses do not restore the original sequence. They reflect an attempt to manage a condition where inputs are present but improperly prepared. Order therefore defines whether inputs become usable outputs. It sets the conditions under which digestion converts food into substrates that can be absorbed and utilized. Changing inputs without restoring order leaves the underlying constraint in place. Restoring order changes how those inputs are processed, allowing the system to operate according to its design and extract value from what is consumed.